We're moving on to chapter 17, which hopefully you're enjoying this journey of learning about chromosomes and what they do. And now we're going to figure out how they turn the genetic data into a protein or a polypeptide. But we hopefully now we all agree that the information on DNA is a specific sequence of nucleotides, that it's inherited, and that being inherited leads to specific traits. The thing that we're going to add in this particular chapter is those traits are dictated by the synthesis of proteins or polypeptides, I might say. And that proteins are the physical link between genotype and phenotype. Proteins are what this DNA is inherited for, what it's passed on for, and what it actually does for us in our bodies is create proteins. So we're going to talk this chapter about the process of creating a protein. It's a step-by-step process. This first image doesn't come from your textbook, but it's very simplistic, which is always good to start out simple. You have your DNA in your nucleus. That DNA will unwind and create a template. That template then will be matched up with messenger RNA. RNA is a molecule that is very similar to DNA, but not the same. But RNA has this special power in the fact that it can leave the nucleus, because DNA can't leave the nucleus. So then messenger RNA can leave the nucleus. That process of DNA in giving its instructions to messenger RNA is called transcription. Then messenger RNA goes out into the cytoplasm and is connected to a ribosome, a free ribosome, and that free ribosome then will bring amino acid sequences that match up with the instructions that the RNA got from the DNA and create this protein. That process is called translation. And this is what we want to learn about here. That's the actual, in a nutshell, what's happening is DNA, which is your genotype, is giving the instructions to create a protein which would manifest your phenotype, what you actually look like, or how you present. The process in which a protein is made is gene expression, and two stages, as I just said: transcription and translation. Transcription in the nucleus, translation in the cytoplasm, and the big key of this machinery is a ribosome, and it's the key piece with regard to, one of the key pieces, there really isn't just one, for polypeptide synthesis. What happens is initially the word enzyme was used. That was deemed to be not efficient enough. Then we went to protein, but the term they've landed on is polypeptide, because polypeptides can get together to form a protein, and officially a protein isn't that until you have a sequence of amino acids into polypeptides. That polypeptide is folded in a certain way, and remember, you can have multiple polypeptides folding together, and then that three-dimensional product that many times has been monitored or changed by the endoplasmic reticulum membrane, is then the protein. But the thing you have here, the other image you have here, is the first image you have of a ribosome. Ribosome is two units. You have a large unit and a small unit. They're kind of like two globs of Play-Doh that are stuck together. You'll see it in this model. You'll also see it in a ribbon model, because the ribbon model is more accurate, I guess, as to what it looks like, because it isn't like Play-Doh. It's more like two globs of yarn stuck together. And in between the two areas where they're stuck together, you have this site, which we call the EPA site, where the messenger RNA is just being read through it. And along the messenger RNA, you have a polypeptide being created. We'll see this again. Going back to our history, remember Mendel was around 1850 to 1870, and then you started out with the precursor to what Watson and Crick did, say around the 20s. At the same time, this man was a medical doctor, Archibald Garrod, was a British guy. Physician, and he worked with metabolic disorders, particularly one metabolic disorder where with the lack of an enzyme, it causes a dramatic change in the color of an individual's urine, turns the urine very, very dark. And then he worked with other metabolic disorders like diabetes, etc. But he just based on his patients, not so much, I mean, he was a busy guy. He was a full-time physician, so he didn't have time for a whole lot of research. But based on his patients, he thought that what was going on with these metabolic disorders that he was working on was the fact that there was an enzyme that they were missing. And so he looked at their family trees, their pedigrees, and looked at the fact that throughout the history of that particular patient's family, you had this particular disorder, this metabolic disorder show up over and over again. So he hypothesized that it was a recessive gene or a recessive trait that was leading these people to the point where they didn't make some particular chemical in the metabolic pathway that was leaving their urine quite dark, kind of blackish. And so he came up with this hypothesis of one gene, one enzyme. He said enzyme, one gene, one enzyme. And he did this around 1909. Nobody really paid any attention to him. He didn't really pursue it as a research scientist. He was a doctor again, very busy man. But it was picked up again by two scientists called Beadle and Tatum. And they confirmed the hypothesis of one gene, one enzyme, but they modified it into one gene, one protein. And they did this with, first of all, they were also working with fruit flies, but the fruit flies were much too complex in their genetic material even so. So they ended up using bread mold. Bread mold has very simple genetic information. It is a perfect specimen because its generations move it along very quickly. So what they did in a nutshell is they took bread mold, they radiated it, and then they knew what the bread mold needed to grow. It needed some sugar, some salt, some amino acids, but the bread mold itself could live with just the sugar and the salt, with just the minimal medium. But what they did was then started eliminating some things from those minimal medium, like they'd eliminate salt, they eliminate sugar, eliminate amino acids. And what they found was it was when the amino acids were eliminated, then the mold did not grow. So then what they did, because there's 20 amino acids, they just systematically eliminated individual amino acids. And they found that this radiated mold that they made, what they did was they made mutant mold was mutated just for one particular amino acid. It was just one amino acid that if it was missing, the mold wouldn't grow. And this was the experiment that really, and of course they were research people, so they published their material around the 1940s. This is the experiment that really solidified the hypothesis that Garrod had worked with, that genes code for proteins. This is more of their data with regard to their experiment, and then going through and eliminating possible amino acids each time they went through to develop this understanding of the hypothesis, one gene, one enzyme. But again, it became one gene, one protein. Because some proteins aren't all enzymes, so later the research one gene, one protein. At that time, it was just enzymes that they were looking at because they were looking at metabolic pathways, but that was why the hypothesis was altered. Now the hypothesis is one gene, one polypeptide, because, as I said, polypeptide isn't necessarily a protein yet, but that actually is what the gene codes for. Genes code for polypeptide chains or RNA molecules, and that was where the exception came up, is genes can code for what eventually becomes a protein, or can code for RNA molecules. And also we've moved on here as we're seeing the transcription. We have that RNA, the messenger RNA, which is going to move out of the nuclear envelope into the cytoplasm where translation is going to take place, and then you have your polypeptide, your groups of amino acids. RNA is the bridge between genes and proteins. Transcription is the synthesis of RNA using the information in DNA. And remember, because a lot of the times when I'm saying these things, that's because that's their definition according to Campbell, according to your textbook. So it becomes a nice little piece of information for you to put in your head for the test eventually as it comes along. And then transcription produces what's known as messenger RNA. We call it mRNA, little mRNA. And then translation is the synthesis of polypeptide using the information on messenger RNA. So these are just explaining what these terms mean. Ribosomes are the site of translation. So ribosomes are in the cytoplasm. They're the site of translation. And you have a nice little image here. This will show up again where you have your RNA. You have an enzyme, RNA polymerase. That RNA polymerase, with the aid of transfer RNA, will attach codons. So the RNA creates codons, will attach these transfer RNAs, will bring amino acids to the codon units on the messenger RNA, and when the transfer RNA brings amino acids to the codon units of the messenger RNA, they're called anticodons. So you have a couple of new terms showing up on this image, which we will define as we go forward, but I do want to make sure there's no confusion in terms on this image that we haven't introduced yet. There is a difference between prokaryotes and eukaryotes, of course. There's many differences between prokaryotes and eukaryotes. The big thing is prokaryotes, translation of messenger RNA can begin before transcription has finished. So remember, prokaryotes, there's no membrane-bound organelles. So this translation, as it's going on and as it is actually sticking out and making a chain of messenger RNA that has been created, that transfer RNA can actually be carrying out transcription. So the polypeptide can be made basically all at once, you might think. Whereas with eukaryotic cells, it's definitely two different processes. You have your transcription, then the messenger RNA needs to be processed meaning looked at, proofread. Messenger RNA then exits the nuclear envelope into the cytoplasm, and then translation can take place in the cytoplasm. We'll pick this up on the next one.